Are you guys fascinated by life? Do you think that the details around us in nature are downright amazing? Do you want to learn more about the world around you? Or do you want a 5 on your AP biology exam? Whether you love nature and biology or just want a 5 on your AP exam, you're in the right place. I'm your host, Belladonna, and welcome to the first episode of That Bio Girl, where I will teach you the key concepts taught in AP Biology so that you can know more about the intricate processes of life inside and outside of you, so that you can get a 5 on your AP Biology exam. Don't worry, it's not that bad. Once you understand the concepts and go one step at a time, I can assure you that what used to be overwhelming for you will not be overwhelming anymore. And I hope that by the end of this, you will walk away with an appreciation for the beauty of life and its processes that are happening all around us. So let's get started. So this first unit is kind of like AP chemistry but simplified. So let's review some intro chemistry. Hopefully you'll remember from intro chem that atoms want to be stable, and the way they make this happen is by forming chemical bonds with other atoms. Think of a chemical bond as a pull or attraction between two atoms that results from sharing or transferring valence electrons. And an important rule to remember here is the octet rule, which posits that elements will gain, lose, or share electrons to complete their valence shell and become stable, to become just like the noble gases, like argon or neon. Then we come across the idea of electronegativity, which, if you remember from intro chem, is basically the measure of an atom's ability to attract electrons to itself. And this, of course, depends on effective nuclear charge and atomic radius, but we don't have to get that specific for this course. So basically, elements have higher electronegativity if they are in the top right corner of the periodic table, because those atoms' nuclei have a stronger pull on their electrons, since the number of protons and electrons is increasing while the number of electron shells is remaining constant. So as you move to the right, or across the periodic table, electronegativity increases while electronegativity decreases as you move down a group on the periodic table. You might be wondering, why are we talking about this? Who cares about electronegativity if we're in AP Bio? Well, electronegativity is an important concept to grasp, since it plays into a very important idea in this unit, the polarity of water. You see, water consists of two hydrogens and one oxygen that are covalently bonded together with polar covalent bonds, which means that electrons are shared unequally between the atoms in any given water molecule. This unequal sharing of electrons creates an imbalance of charge and results in partial charges on the oxygen and the hydrogens. Since oxygen is more electronegative, it has a stronger pull on the hydrogen's electrons because it is greedier to fill its valence electron shell. As a result, those hydrogens have partially positive charges, and water's imbalance of charge allows for the formation of hydrogen bonds, which are the key to water's special properties that it provides for life. Hydrogen bonds occur when a partially positive charged hydrogen atom that is already bonded to an electronegative atom, like oxygen, nitrogen, or fluorine, bonds with an electronegative atom on another molecule that has a partially negative charge. Let's use water molecules as an example. So since water has an imbalance of charges due to its polar intramolecular, meaning inside one molecule, covalent bonds, it has a partially positive hydrogen already attached to an electronegative oxygen. And when a hydrogen on one water molecule interacts with an electronegative oxygen atom on another water molecule, a hydrogen bond is formed, since the partially positive is attracted to the partially negative. Okay. So I kind of skipped over ionic bonds and nonpolar covalent bonds, so I'm going to quickly circle back to those now. If you remember from intro chem, ionic bonds are attractions between oppositely charged atoms called ions, and they usually occur between a metal and a nonmetal. And some examples include NaCl or sodium chloride and LiF or lithium fluoride, where sodium and lithium are the cations, or positively charged ions, and chloride and fluorine are the negatively charged ions. And these positively and negatively charged particles form ionic compounds and crystal lattice structures. And in the same way that hydrogen in water can bond to an electronegative atom in another water molecule, in another molecule in general, the hydrogen and oxygen in water can also form attractions with ionic compounds, like when sodium chloride dissolves in water. The partially positive hydrogen in water is attracted to the partially negative chloride, and the partially positive sodium is attracted to the partially negative oxygen and form ionic bonds. And then the last kind of bonds that I'll review here are ionic bonds. Are covalent bonds, which I touched on earlier, but I am going to explain now. They occur when two or more non-metal atoms share electrons. And they can form single, double, or triple bonds, which are each pairs of shared electrons. A single bond is one pair of shared electrons, a double bond is two pairs of shared electrons, etc. And this will be important later when we discuss how carbon can single, double, and triple bond to itself in macromolecules. Covalent bonds can either be nonpolar covalent or polar covalent, and it is very important to note that nonpolar covalent bonds are hydrophobic and thus nonpolar, since their electrons are shared equally between the atoms. Examples of nonpolar covalent bonds include O2 or CO2, and we already discussed that polar covalent bonds are where electrons are shared unequally, which creates a dipole, which is another word for a polar molecule. So now that we've reviewed some intro chemistry, let's dive into the properties of water. The main properties of water that you should know are water's polarity, cohesion, adhesion, capillary action, temperature control, and density. Let's start with water's polarity and cohesion. Like I said earlier, water has a partially negative side and a partially positive side, which allows for hydrogen bonding. And hydrogen bonding enables all of the following special properties of water, like cohesion, which is the attraction of molecules for other molecules of the same kind. In other words, this means that water is attracted to itself, since it can form hydrogen bonds with itself, which is very important for life because water's hydrogen bonds within its water molecules allow water molecules to move up together against gravity, which allows for the transport of nutrients in plants, for example. You may also see cohesion and surface tension used interchangeably, since cohesion is responsible for surface tension, since hydrogen bonds between water molecules are pretty strong and allow water molecules to stick together for a long time before breaking. The next interrelated property of water is adhesion, which describes where water is attracted to other polar molecules other than itself, which is very important in plants, because water molecules can cling to the molecules of the cell walls and the xylem, the plant vascular tissue, which is polar, and resist the downward pull of gravity that way. The next property of water is capillary action, which combines adhesion and cohesion into one property, the upward movement of water due to the forces of cohesion, adhesion, and surface tension. For capillary action to take place, adhesion has to logically be greater than cohesion, otherwise the water would not move up the plant. And for this property, again, just remember that this is very, very important for the transfer of water and nutrients in plants, and can happen all because of those hydrogen bonds. Water's next property is also very important, and it is the property of temperature control. And one of the main facets of this property is high specific heat, which means that water is able to absorb a lot of heat energy before breaking due to its hydrogen bonds. In other words, water resists changes in temperature to itself as it takes in heat, which means that large bodies of water like oceans can absorb a lot of heat in the daytime and release it at night, thus stabilizing the external temperature. Now you may be wondering, how in the world can water do that? Well, it's able to absorb heat from the air and cool the air because it does not evaporate quickly because of its high heat of vaporization, which basically means that water has to absorb a lot of heat energy before it vaporizes or turns into a gas from a liquid. Now you may be wondering, how can it absorb a lot of heat energy while other chemicals can't? Once again, what do you think is going to come to the rescue? Obviously, hydrogen bonds. Water's large number of hydrogen bonds in a large body of water give it great stability. because hydrogen bonds are quite strong intermolecular forces. As a result of the hydrogen bond's collective strength, more heat energy is needed to break water's hydrogen bonds and vaporize it. Hence, oceans are able to absorb lots of heat and cool the surrounding air off while not evaporating or experiencing changes in its internal temperature. Water's high specific heat is very important for animals too, since organisms with water inside of them can resist changes in their own internal temperature, since the water within them absorbs heat energy that could otherwise alter the organism's internal temperature. While I did touch on water's resistance to evaporation earlier, it ties into water's property of evaporative cooling, which explains that water does evaporate after it has reached a certain point of absorbing a lot of heat energy. However, since evaporation is an endothermic process, meaning that water needs to absorb, not release, a lot of heat energy from the surrounding air for its hydrogen bonds to break, water cools the temperature of the atmosphere when it becomes a gas, which moderates Earth's climate, stabilizes the temperature in lakes and ponds, prevents organisms from overheating, and prevents leaves from becoming too warm in the sun. The last property of water that relates to life is water's density, which means that as water solidifies, it expands and becomes less dense, since extra hydrogen bonds that form a crystal lattice when water freezes increase space between molecules, which decreases density and causes ice to float, which is so important since it allows marine life to survive under floating ice sheets in the aquatic world below, which I'm thinking about right now because it actually just snowed a little bit today, so I'm sure that some bodies of water nearby are frozen over and there's aquatic life that's able to survive below. Can you imagine if water was not able to hydrogen bond in this scenario? The ice would sink to the bottom of whatever large body of water it formed on, crushing aquatic life, and water would not be able to transport nutrients in plants, absorb heat energy, and cool the air down if it were not for hydrogen bonds. So I said that water's density is its last property relating to life or the environmental world, but chemically speaking, water is also a universal solvent, or a dissolving agent, in a solution because of its polarity. And some words that you should know surrounding solutions in general are a solution, which is a homogeneous mix of two or more substances, a solute, which is anything that dissolves, while the solvent, in this case water, is the substance that does the dissolving. In water's case, its polar molecules are attracted to ions and other polar molecules that can form hydrogen bonds with, so it is able to dissolve polar molecules like sugars or proteins containing many oxygens and hydrogens and ions, which are all the solutes, by forming hydrogen bonds with those substances in a similar way that it does with ionic compounds. And just a quick note, when I was talking earlier about how water forms bonds with sodium and chloride, just note that anions are ions with negatively charged particles. I did not remember to say that earlier, so I just wanted to drop that right now. The anions are negatively charged ions and cations are positively charged ions. But back to dissolving. An important distinction to make is that polar molecules do not dissolve nonpolar molecules because nonpolar molecules are hydrophobic and do not like water. You can also think of their no love but all hate relationship as being because nonpolar molecules like diatomic oxygen physically cannot form the necessary hydrogen bonds, since the only intermolecular bonds they can form are London dispersion forces, not hydrogen bonds. It's like when you go on your calculator and try to add two matrices with different dimensions and you get a dimension mismatch message from your calculator. In the same way that you can't add two matrices with different dimensions, you can't add a molecule that can only form London dispersion forces with a molecule that forms hydrogen bonds. You may not get that, but take with it what you will. Okay, we've talked a lot about water, but now let's talk about the main bulk of this unit, organic compounds, which are compounds that contain carbon and hydrogen, and a little bit of oxygen, of course, but with the exception of CO2 since it's too small to be considered an organic compound. Like I hinted at earlier, carbon can form single, double, or triple covalent bonds, and a single carbon can form up to 4 covalent bonds and can form long chains and rings. But keep in mind that the type and number of covalent bonds carbon forms with other atoms affects the length of the carbon chain and thus the shape of the molecule that will be created. Since a single bond is long, a double bond is shorter, and a triple bond has the shortest length. Going back to carbon chains, which are just links of carbon atoms, hydrocarbons can also be formed, which are organic molecules consisting only of carbon and hydrogen, and these are important because they are the framework, or the backbone, for more complex organic molecules. But again, it's important to note that the sizes, lengths, branching abilities, double bond positions, and presence of rings all vary for each hydrocarbon backbone of any given molecule. While hydrocarbons are important for forming a backbone of a molecule, there are also functional groups on molecules too, which are chemical groups attached to the carbon skeleton that participate in chemical reactions. And if you can, I would definitely try to memorize these if you want an edge on the exam. The main functional groups to know are hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, methyl, and phosphate groups. And all of those are polar groups except the methyl group, which is nonpolar. Knowing these functional groups is important since it will give you an indication of how that particular part of a complex molecule will behave in the presence of nonpolar and polar molecules. Okay, now that we know some basics about structure, we can talk about macromolecules, which are organic compounds that are key to life and consist of carbohydrates, proteins, nucleic acids, and lipids. Before we get into specifics about each of these groups, though, let's talk about the formation and breakdown of these macromolecules. To form a polymer, which is a chain-like macromolecule of similar or identical repeating units covalently bonded together, we'll need a dehydration reaction, which bonds two monomers, which are the repeating units found in polymers, with the loss of water. In reality, what happens is that the hydroxide, or OH, of one monomer bonds to the hydrogen of another monomer, forming water that is then released. So basically, in this process, we're building a large molecule with the removal of water. And this process is endothermic, since it requires energy to take place. It's also important to note that a dehydration synthesis reaction connects every single monomer to itself in the world of macromolecules. On the other hand, hydrolysis is the opposite. It involves the breaking of bonds in a polymer by adding water. So one hydrogen of the water molecule bonds to one monomer, and the remaining hydroxide of the H2O attaches to the other monomer, thus breaking the larger molecule down by adding water. Don't forget that hydrolysis releases energy, so it is an exothermic reaction. Okay, now that we know how polymers are broken down and formed, let's talk about carbohydrates, which are made of carbon, hydrogen, and oxygen. Carbohydrates contain a carbonyl group and many hydroxyl groups, and since these are both polar functional groups, hint hint there, the entire molecule is at least somewhat polar and therefore hydrophilic. Remember what I said earlier about monomers? Well, monosaccharides are the monomers of carbohydrates. They're simple sugars that form glycosidic bonds when they join together. The most common monosaccharide is glucose, which is important in cellular respiration, very important, and is a nutrient and fuel in cells. Another monosaccharide is fructose, which is a sugar that comprises a fruit like an apple, and it is an isomer of glucose, which just means that both molecules have the same chemical formula but have a different chemical structure or arrangement of atoms. These monosaccharides are very important because they serve as building blocks for disaccharides and polysaccharides. Disaccharides are two monosaccharides joined together with covalent bonds, or the glycosidic bonds I mentioned just a second ago. And a disaccharide is not exactly a polymer of a carbohydrate, but it is sort of an in-between molecule. It's not a true monomer, but not necessarily a true polymer either. The most common disaccharide is sucrose, though, which is formed when glucose and fructose join together. Of course, with a dehydration reaction. Sucrose is important since plants transfer carbohydrates from the roots to their leaves in the form of sucrose. Now, polysaccharides, or polysacs for this podcast purposes, don't say that on your exam, just for this podcast, are the main polymers of carbohydrates with hundreds of sugars joined via dehydration reactions with glycosidic linkage. There are two main types of polysacs to remember: storage polysacs and structural polysacs. Storage polysacs consist of starch and glycogen. The only difference between these two forms of excess glucose are that plants store starch and animals store glycogen in their liver and muscle cells. Lastly, the structural polysacs consist of cellulose and chitin. Cellulose is a tough substance that forms plant cell walls, and chitin, which contains nitrogen, forms the exoskeleton of arthropods. Now let's talk about proteins. They are the most diverse and abundant macromolecules with so many functions that we can't get into them all. A protein is a molecule, or macromolecule to be specific, consisting of polypeptides, which are the polymers of proteins that are folded into a 3D shape. Proteins are usually made up of carbon, hydrogen, oxygen, nitrogen, and sulfur. And keep an eye on what elements comprise these macromolecules, because you may get a question that tests your knowledge on the macromolecule's composition. The very important thing to remember here with proteins is that shape determines function. They are very specifically shaped depending on what function they have. Now for proteins' monomers. Proteins' monomers are amino acids, and 20 different amino acids exist in nature. They each have an amino group, a carboxyl group, an alpha carbon in the center of the amino acid, and an R group, which is a variable side chain with distinct physical and chemical properties that give the amino acid its distinct properties like polarity. The R groups can be characterized as either nonpolar, hydrophobic, polar or hydrophilic, or charged or ionic, which is counted as hydrophilic. These R group side chains interact, and depending on their polarities, they determine the unique shape and structure of the protein, which determines its function. The side chain interactions are very important to note because the R groups determine whether the amino acid is hydrophobic or hydrophilic. And if an amino acid is hydrophobic, it will orient itself away from water in a protein structure. But if it is hydrophilic, it will orient itself towards water in a protein structure. So keep that in mind. Now, to connect amino acids, we have to form a peptide bond. And in order for that to happen, the carboxyl group of one amino acid must be next to the amino group of another amino acid. Then, via a dehydration synthesis reaction, obviously, a peptide bond forms. Once a long string of amino acids exists, we have a polypeptide, a string of amino acids linked by peptide bonds. And on each end of any given polypeptide, note that there is a free amino group called the N-terminus, and on the other end there is a free carboxyl group called the C-terminus. Now we've talked about the sequence of amino acids and how it determines the 3D shape, but let's talk now about what happens when a polypeptide twists and folds from those R group interactions to form a protein. Proteins have 4 levels of structure: primary, secondary, tertiary, and quaternary. In primary structure, there's just a linear chain of amino acids that is determined by the genes solely. Then in secondary structure, the string of amino acids begins to coil and fold due to hydrogen bonding that occurs between different parts of the amino acid string, like between the carboxyl group in one peptide bond and the amino group in another. The coils and folds that contribute to secondary structure can either result in an alpha helix structure or a beta pleated structure, and those are both very important to note, what kinds of structure a protein has in secondary structure. Then in tertiary structure, 3D folding starts to occur between the side chains of amino acids. So for example, certain polar R groups may form interactions with other polar R groups, and the same goes for nonpolar R groups. Lastly, quaternary structure is the linkage of 2 or more polypeptides that have slightly different properties when they join together, called emergent properties, but this structure is only found in some proteins, like hemoglobin in our blood. Based on these different structures, proteins can have many functions in the human body. They can be antibodies, which help protect the body from disease, enzymes, which we will discuss a lot more in Unit 3, messengers that transmit signals like hormones in cell communication, which we'll talk about in Unit 4, structural support in muscles and bones, and transporting molecules like hemoglobin that transport small atoms and molecules throughout the body. Now let's discuss nucleic acids, which consist of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Nucleic acids' function is to store, transmit, and express an organism's hereditary information, and they can either be DNA or RNA. Nucleotides are the monomers of nucleic acids, and they each consist of a phosphate group, a 5-carbon sugar, and a nitrogenous base. The phosphate group component is added to the 5-carbon sugar to form the nucleotide, and the 5-carbon sugar component is bonded to the nitrogenous base in the nucleotide. And this sugar can either be deoxyribose if it's DNA or ribose if it's RNA. The difference between these two sugars is that ribose has an extra hydroxyl group that deoxyribose lacks. Lastly, on a nucleotide, we have the nitrogenous bases, which can either be pyrimidines or purines. Pyrimidines have one ring with 6 atoms and include cytosine, thymine for DNA, and uracil for RNA, and purines have one ring with 6 atoms bonded to one ring with 5 atoms, so they have a double ring. And they include adenine and guanine, both of which are universal for both RNA and DNA. This will be especially important later, but remember that adenine pairs with thymine and cytosine pairs with guanine in DNA, and adenine pairs with uracil, while cytosine pairs with guanine in RNA. Now that we've unpacked the details of the monomers of nucleic acids, we can discuss the polymers of nucleic acids, called polynucleotides. And it's important to note that structurally, phosphate groups link adjacent nucleotides and connect via bonds called phosphodiester linkage via a dehydration reaction, obviously. Polynucleotides also have a certain directionality. For DNA, one side is 5' to 3', meaning that the 5' end is the phosphate end at the top, and the 3' end is the hydroxyl end at the bottom, and the other side is 3' to 5', meaning that the hydroxyl end is at the top and the 5' end is the phosphate group on the bottom. Because DNA has 2 strands, it forms a double helix structure where the strands are anti-parallel. And anti-parallel just means running in opposite directions. And this is a striking characteristic of DNA. Additionally, note that the base pairs in DNA that I mentioned earlier, like cytosine with guanine and adenine with thymine, are held together by hydrogen bonds, which may be strong but are still able to easily break apart for DNA replication. So they may be able to break apart for DNA replication, but still note that they're pretty strong when it comes to water. If you're confused about DNA's anti-parallel strands a little bit, don't worry. We'll dive into DNA structure a lot more when we discuss DNA replication in Unit 6. As for RNA, shifting from DNA, it is a single-stranded polynucleotide that is variable in shape but always has adenine pair with uracil while cytosine pairs with guanine. Additionally, note that the specific sequence of bases along DNA or RNA is unique for each gene and dictates the amino acid sequence or primary structure of the protein that I talked about a few minutes ago. Now, before we get to lipids, our last group of macromolecules and the conclusion of episode 1 of That Bio Girl, I want to let that fact about which bases pair with which bases sink in. If you are asked on the AP exam to provide the complementary strand for a piece of DNA, remember, please remember that cytosine goes with guanine and thymine goes with adenine. If you are asked for the RNA strand complement, please remember that adenine goes with uracil and cytosine goes with guanine. Please do not forget that. Now we are on the last group of macromolecules, the lipids, which mainly consist of carbon and hydrogen, but also contain a little bit of oxygen and certainly phosphorus in phospholipids. Lipids are a class of molecules that do not include true polymers, and they are generally small in size, so they actually are sometimes not considered to be macromolecules, but here we are calling lipids macromolecules because they're important. The types of lipids are fats, phospholipids, and steroids, and they are hydrophobic because they are nonpolar. Fats are composed of glycerol and fatty acids, which are long carbon chains with a carboxyl group at one end. To form a fat, 3 fatty acids are joined to a glycerol via a dehydration synthesis reaction and form an ester linkage, a bond between the hydroxyl and carboxyl group. As for fatty acids themselves, they can either be classified as a saturated fatty acid or an unsaturated fatty acid. A saturated fatty acid has no double bonds between carbons in the carbon chain, which means that there are more hydrogens present, and they are solid at room temperature and include substances like animal fat or butter. And an unsaturated fatty acid contains one or more double bonds, and it has kinks within itself that cause it to be a liquid at room temperature. Examples of an unsaturated fatty acid include olive oil and the healthy oils in general. In addition to fats, phospholipids are also important lipids, and they are the major component of cell membranes and consist of 2 fatty acids attached to a glycerol and a phosphate. These phospholipids assemble as a bilayer in water, which means that there is one layer of phospholipids whose heads are orienting towards the extracellular aqueous environment, while there's another layer of phospholipids whose heads are orienting towards the intracellular aqueous environment, leaving the fatty acid tails orienting themselves away from the aqueous environments by comprising the middle nonpolar section of the cell membrane. Going back to fatty acid tails, while it may seem trivial, it is important to note when an organism has either saturated or unsaturated fatty acids in its cell membrane, since in Unit 2, you will see that it is advantageous for an organism to have either saturated or unsaturated fatty acid tails in their phospholipid bilayer in their cell membranes depending on the environment that they live in. Regardless of their saturation, though, the fatty acid tails are always hydrophobic, and the phospholipid head is hydrophilic. So in the cell membrane, the tails orient themselves away from the aqueous environment, while the heads orient themselves towards the aqueous environment, both intracellular and extracellular. Lastly, steroids are lipids that have 4 fused rings, and the functional groups attached to their rings determine the type of steroid that a given steroid is. Testosterone and estrogen are great examples of very important nonpolar hormones that are lipids. Okay guys, I know that was a lot of information, but you stuck through with it, and I really hope that episode 1 of That Bio Girl has given you a better grasp on Unit 1, the chemistry of life in AP Biology, and I hope you walked away with a little more appreciation for the complexities in the biological world around us. Feel free to like this episode, tell your friends about it, and check out our Instagram page called That Bio Girl, where you can find out when each new episode will drop. See you next time, and don't forget to dream big.